thanks a lot for the opportunity to show up our recent studies on high-throughput phenotyping. Well, I will go to introduce how we evaluate the adaptability of Norway spruce seedlings to drought stress by merging multisensory high-throughput phenotyping technique with different omics. Well, first, I would like to mention that it's very important forest in Austria. As you might see here in these pictures, 47% or 48% of the surface of the country is covered by forest and is one of the main industry of the country. Actually, spruce itself is the most important species which is it is produced, and the forest in total represents 6% of Austrian economy. So therefore, the spruce is representing actually 3 4 of the Austrian economy. But although this is a distribution, the current distribution of spruce in Europe, it's a very important tree because it's mainly used for pulp production and wood production. Nowadays, we know that potential future distribution will strongly change due to the climate change. And due to the fact that drought stress is one of the major causes of drought, of stress for Norway spruce, because of the root architecture and so on, we are very much interested to see whether we could find some diversity on how to perform against the drought. For this reason, before having any kind of high throughput technology, we were working mainly based on provenance trials, which means you have different provenances which were planted decades ago in a specific spot. So therefore you control, let's say, for the same conditions, and therefore you go to this spot and you go tree by tree by taking cores, like 3 cores from them, and you could then study by X-ray how was the growth of the tree in a specific year. Well, this is a very nice image and analysis that allows you to phenotype, but it's very expensive and time consuming. And anyway, this allows you to get some nice traits that are commonly used in drought stress, like resistance, which is comparing the drought stress growth to the pre-drought stress growth. When a plant grows better in a drought stress than or before a drought stress, it means that is even coping better than being just resistant. Well, we did a publication some years ago by showing that there was somehow a diversity on the response to drought, and we were even able to associate some SNPs to this diversity and these traits. But of course, it was in a very limited and narrow set of genetic diversity, because as I told you, we were working with a provenance trial, and even you don't have an appropriate experimental design that was designed 20 or 30 years ago, or it's very hard nowadays to establish new sets of experiments and then wait until the trees are growing. Therefore, I was in deep discussions with Jakub at the time that he was together with his team designing the new high-throughput phenotyping facility in Vienna. And we actually asked for a project to be able to work on this. The idea was to reduce the time and to be able to include as much as provenances as we can by using seedlings of a spruce in a way that we can find differences among the different germplasm material. I will give now the voice to Ahmad, who was the postdoc that we hired for this project.
So hello everyone, my name is Ahmad, as Carlos introduced me. I am working as a postdoc at the Austrian Research Centre for Forest, and I was mainly hired for investigating the drought stress tolerance or adaptation in spruce populations. So like 1.5 years ago, we started to use Jakub phenotyping facility to understand drought stress responses. And the first experiment basically we wanted to find out whether this system would be useful in screening different populations. So at the time when we started, we only have one population of spruce. And then what we did, basically, we grow them at different water potentials, which represent different kinds of different levels of water stress. And what you could see here on the images, under different water potential, we could see a nice gradient in the chlorophyll fluorescence data, because as you move from left to right, you could see that as the more active the water potential is, the lower the chlorophyll fluorescence is, meaning that the plants, you can nicely differentiate between different gradients of a drought stress. And this was also the case from the second sensor, which is basically coming from shortwave infrared region, and which actually act as a proxy of tissue water contents, where we see that the plants which are growing on the lowest water potential are basically has a very lower tissue water contents as compared to the other treatments. So this gives us a proof of concept that the system can work either even though we were screening different stress levels, but not different populations. Then it was also, we looked at how this performed across the time points, and what we see along the time points, the drought severity, not in terms of treatment, but also along the time points, you could see that you can finally capture the temporal dynamics of drought stress. So to show that whether this could be useful when we are screening different populations, we set up a different experiment where we sampled two different populations of Norway spruce. One was coming from higher altitude, where the region is basically, climate is basically characterized by high rainfall and the lower temperature. And another population basically was originating from a climate which is relatively dry and warmer. So we did a 21 days drought stress experiment where we grow the populations for 8 weeks under normal conditions, and then we we give them the drought stress for 3 weeks, and we do high throughput phenotyping based on 4 different sensors. One was RGB, chlorophyll fluorescence, and hyperspectral. And hyperspectral has 2 ranges, one coming from the shortwave infrared region, and the second one from visible near infrared regions. What additional we did basically along this experimental drought phase, we sampled plant tissues for metabolomics analysis, for RNA-seq and anatomical analysis, to understand how phenotypic data basically match those data sets as well. So to give as a proof of concept that the technology works and it's in congruence with each other. So from phenotyping, from all these sensors, from RGB, we basically estimated the plant growth. From chlorophyll fluorescence imaging data, we were able to estimate the photosynthetic ability of the different plants. And from hyperspectral data, we calculated different vegetation indices, which tells you the vegetation health. And in total, we had around 56 different traits, which we captured using all these all different sensors. And when you put all these different traits into a principal component, it was very nice to see as a first look that your treatments, which are represented here by different symbols, are splitting along the first axis, which is expected. But what we also see that along the second axis, the temporal component, meaning that when the drought stress is becoming more and more severe, the samples are splitting based on the drought stress severity. So there is also, we could nicely capture the temporal component of the drought stress by using all these sensors. But what was more surprising for us, or also kind of expected, because we wanted to see differences between populations. And when we plot the third principal component, we could nicely see that on the drought axis, that the populations could also be splitted based on these traits. So basically, when we looked into one of the component, which is a time, which is interesting, we saw that there are many traits which were captured by the time component, but what was more interesting for us is the non-photochemical quenching, which basically is a mechanism by which plant dissipate excess heat when you are under drought stress. Plant cannot use all of the light, and this excess light is basically dissipated as a heat, which is, we called it as a non-photochemical quenching. So this was captured nicely on the time component. And when we plot this trait for both populations, what we could see that in the initial phase of the drought, the non-photochemical quenching is increasing, meaning that plant is able to cope with the drought stress. But as long as we move along the drought stress, this ability of the plant is going to decline, meaning that then the drought stress is so severe that the plant is not able to cope any further to the drought stress. And what we also see is that there was temporal differences, how there is an activation of non-photochemical quenching between different populations. So non-photochemical quenching basically work with the increase in certain metabolites, for example, zeaxanthin. And in parallel, what we see that this compound was also increased under drought stress, so meaning that both data sets were highly supporting each other. And then we performed the metabolomics analysis. In this way, we did untargeted metabolomics using LCMS. And again, what we see very clearly is that when you look all the data sets and make a principal component, the treatments are being separated on the first axis along with the time. As the drought become more and more severe, there is a much more changes in the metabolites profiles, and the samples are much more separated. But what was interesting for us to see that there was not only separation between populations under drought stress condition on the second axis, but we also see constitutive differences in the metabolic profiles along the second axis. So meaning that the both populations differ in terms of their metabolic profiles, which also fit with the phenomic data set. When we look into all these metabolites, what we see clearly that there were three different clusters. One cluster is basically the one which increase upon drought stress. The second cluster is the one which basically goes down, those metabolites which downregulated. But there was the final cluster, which is the cluster number 3, which differentiate the two provenances, because here these clusters, these metabolites are much more abundant in one population as compared to the second one. Then we were interested in RNA-seq. So what is happening at the transcriptional level? So we did RNA-seq at 14 days after drought stress. And again, we saw the same picture that the treatment are being separated along the first axis, and our populations were splitting along the second axis, and giving us a nice complementation of the metabolomics and the phenomics data set. Lastly, we did anatomical analysis, from the stem sections. So we were interested to understand how the stem anatomy is changing under drought stress, and whether there are differences between populations. And what we see here again, that the drought stress severely affect the plant stem. As you could see, the size of the stem has reduced in both populations. But what was interesting is that the xylem, as you could see in the pie charts, that the proportion of the xylem tissues was much more significantly affected in one population as compared to the second population. And it means that under drought stress, the xylem was heavily affected in both populations, but one population has a more significant effect as compared to the other one, showing the differences between the populations. So overall, if I sum up the whole data set or whole presentation, what we clearly see is that what we see in the phenomics data, which allows us to distinguish between all three factors of our experiment, which is treatment, populations, and the time. This was also captured by both the metabolomics data set and, of course, on one time point with RNA-seq data set. So this gives us a proof of concept that the technology works, which is highly in complement with different analyses. So we now just recently finished a large-scale experiment using the same high-throughput phenotyping setup. We screened 71 populations of spruce from all over the distribution, and the analysis of this is now under progress. So overall, the experiment went quite well, and we hope to see much more differences between populations than what we saw in the current experiment. Thank you very much. And at the end, I would like to acknowledge all my colleagues and our funding agency, which is Bundesministerium. So thank you very much.
Thank you very much for inviting me. My name is Jakub. I'm heading the Plant Sciences facility at the Vienna BioCenter core facilities. Yeah, thanks for the great presentation, Ahmad and Carlos. As we were designing the infrastructure 5 years ago, I have never thought and encountered that we will also run trees in our installation. But as you have seen, the infrastructure is also compatible with trees, obviously, also with small needle trees, which is very, very interesting for us to see. So I will show you today in 6 slides and 6 minutes, my last 6 years, basically. So we are one of 11 scientific core facilities at the Vienna BioCenter, providing access to state-of-the-art research infrastructure to the researchers on campus, but also beyond. And we are also in charge of technological innovations by designing, building, and also implementing of novel technologies, instrumentations, but also research infrastructures. And so we did also with our newest research infrastructure. And this is PhenoPLANT, which was partially presented by Ahmad before. So it's the Austria's first and only high throughput and multi-sensor plant phenotyping infrastructure, which is fully incorporated into a huge phytotron. So this huge phytotron gives us the advantage of having super precise control of environmental conditions like temperature, humidity, light, and also CO2 levels. So what we do here is we try to disentangle the G/E interaction triangle that you see on the right side. So we submit samples with a certain genotype to our system. We control the environment. We apply abiotic or biotic stresses. And we do a non-invasive phenotyping. So on the right side, you see the conveyor belts where the genotypes, the plants, the samples are growing under highly controlled environments. And on a daily basis, they are basically moving to the phytotron on the left-hand side of this slide and moving through the imaging boxes, and every imaging box hosts another sensor. The plants are moving out in the end and are submitted to a gravimetric and fully automated watering system that allows us basically the full control over the water content of the pot, over the water potential, which again gives us full control and highly precise control, for example, for drought stress experiments. But on the other hand, this automated water application system can also be utilized for application of biostimulants, pesticides, nutrients, salt stress, and other liquids agents. And then after imaging, the plants move out again to the so-called waiting room and grow on and wait for the next measurement. So the environment is super highly precise controlled, plus minus 0.2 degrees, between 0 and 40 degrees. So some heat stress is possible up to 40 degrees and some cold stress down to 0 degrees. We have a full humidity control. We can control the CO2. So we can simulate atmospheres, CO2 atmospheres that are predicted to happen in the next 100 or 200 years or even beyond. The plants are growing under state-of-the-art multispectral LED panel where we have the full control of different LED channels. So we can simulate not only different intensities up to highlight and more than 1,300 micromoles, but also we can work on the spectrum and set different spectral conditions, like, for example, romantic red sunsets. But the system can also simulate clouds passing by if this is requested. So the one point is highly precise control of the environment for really sensitive experiments. But what we can also do is we can do dynamic environmental simulations. So we can do it even live, but we can submit data from other environmental stations and, for example, simulate, I don't know, a Spanish summer or a Swedish spring. This would be also possible in our installation. The other phytotron hosts the sensors. We included as many as we could afford, and I think they are all sensors that are meaningful for plant phenotyping, for high throughput plant phenotyping. We have RGB imaging from the top, RGB from the side. Everything is on turntables, so we do multi-view imaging. We have a chlorophyll fluorescence cam for chlorophyll fluorescent imaging and for the investigation of the PS2 system, which is a very nice indicator for plant stress. We have thermal imaging on board, a 3D laser scanner for 3D reconstructions of the canopy, which gives you information on the 3D biomass, leaf inclination, and other 3D information. And we have two high resolution hyperspectral cams that Ahmad was talking about, where we can score tons of vegetational indices, but also the chlorophyll content, the water content, nitrogen content, anthocyanin content, and do really a non-invasive spectroscopy on the plant leaf. The system was designed basically not for me, but for you. So we considered in the design to be able to submit as many different sample types as possible. So we can submit multi-well agar plates for basic research, for example, like we did with Macantia or Arabidopsis. We have small pots for Arabidopsis, but we can go up to 5-liter pots and host also small trees or crop plants up to 70 centimeters, like barley, wheat, and other crop plant species like camelina. And that's my last slide. The next slide is my last slide. I would like to show you a very quick and short video of one of our last experiments, where you basically get the point of view of a sample passing our system. Enjoy.
Thank you so much. It's an impressive lab. Actually, I think it's quite unique to have this all environment settings.
There's RGB top view, RGB side view, one turn, another side view, a 3D triangulation laser scanner, capturing the 3D information from 6 sides, and 2 hyperspec scanning cams. And then the plants move out to the gravimetric watering units. The weight is being scored, and the proper water amount is applied. And the plants are going back to the waiting room, waiting for another measurement. Yeah, so the VBCF and so our facility is part of the Austrian BioImaging node, the CMI. The homepage is listed here. Please visit the homepage for more information on the technologies available in Austria. Yeah, and I would just like to point your attention, like was done in the very beginning, to the funded access to many interesting research infrastructures, and so also to PhenoPLANT via the AgroSurf project. And I would like to thank you for your attention.